Today we are going to be talking about the instrumentation that is used in a microbiology lab. The instrument used in the microbiology lab includes a bunch of different varieties of instruments with their working on varied sophisticated technique with advancing technology, which provides the ample opportunities to make the laboratory testing more accurate, fast, and reliable. So if the test results are to be useful to the doctors in the diagnosis and treatment of disease, the test must be performed as accurately as possible with short testing time. So this requires the use of good instrumentation and of sound analytical methods. Since there are varieties of instrument, we are going to be discussing only the common one that we use. So the first one we have here is an analytical balance. This analytical balance is a type of balance that is commonly used for the measurement of mass in the sub-milligram range. As you can see here in the given image, this analytical balance um, it is made up of a measuring pan, and it is enclosed with a transparent cover. So this transparent cover prevents the small particles or air currents from getting collected on the pan, and this electrical analytical balance, they uses the force necessary to counteract the mass rather than measuring the mass itself. So this um, this analytical balance also create a force required to achieve a balance with the mass or the substance, and the resulting force is displayed. So as they are highly precise and based on the advanced technology, this analytical balance are used in laboratories for the effective completion of tasks like weighing the test materials, then formulation, then for the density determination, then for the quality control testing, etc. So next we have here is the autoclave. So this autoclave is a moist heat sterilization. It is used for the process of sterilization by combining the three factors: time, pressure, and steam. So the way this autoclave work is that it uses the steam as the sterilization agent, and the main principle of this autoclave is that all the items within the autoclave come in direct contact with the steam for a particular period, irrespective of the nature of the material, whether it is plastic, glassware, or some liquids. And also these autoclaves are mostly used for the sterilization of medical laboratory equipment, and they are commonly used for preparing the culture media, which is used for growing the microorganism. Next we have here is the hot air oven. This hot air oven is a type of a dry heat sterilization, and it is performed on the dry materials only. And also there are two types of hot air oven based on their working principle. So the first one is the forced air hot air oven. In this type of hot air oven, the heated air inside the oven is distributed throughout the oven with a fan, and it prevents the rising of a hot air towards the top while keeping the cold air at the bottom, and this allows the adequate heating of the material inside the oven. The next we have is the static air hot air oven. So in this type of oven, the heat is produced by coils present at the bottom of the oven with no fan, and the hot air rises and does not allow the effective sterilization of the material. So this hot air oven can be used to sterilize material like glassware, metal equipments, etc., but not the plastic, and it also allows for the destruction of microorganism as well as the bacterial spores. Next we have is the incubator. So this incubator is a device that is used in the laboratories for the growth and the maintenance of the microorganism and culture. The way this incubator work is based on the principle of maintaining a proper atmosphere for the growth of microorganism, and similarly They are provided with adjustment for maintaining the concentration of carbon dioxide and to maintain the pH and humidity which is required for the growth of the organism. These incubators also have a wide range of they also have a wide range of applications, including the cell cultures, pharmaceutical studies, hematological studies, and the biochemical studies. Next we have is the centrifuge. So this centrifuge is a device that allows the rotation of an object about a single axis where an the outer force is applied perpendicular to the axis, and this centrifuge is a motor base and allows the rotation of a liquid sample, which results in separation of the component of the mixture. So the way this centrifuge work is that it works on the principle of the sedimentation with the high speed of the rotation, which causes the denser particle to settle at the bottom while the lighter particles are collected at the top. So in the laboratory, the tabletop centrifuge, the sample tubes are aligned at an angle so that the particle have to travel a shorter distance before they hit the bottom. So this centrifuge, it can be used for the separation of cell organelles, nucleic acid, blood components, and also for the separation of the isotopes. So next we have here is the microscope, which is commonly used and very necessary in the laboratory. So this microscope is a device that allows the observer to see this smallest particle that cannot be seen with our naked eye. And there are many types of microscope. Each of these works on their own respective principle, but the basic principle of this microscope is magnification. So different types of microscope are developed to cater the specific need of the observation. So based on the different types of different microscope are used for different purposes, and they are primarily used for the observation of minute particles that cannot be seen with our naked eye. Next we have is the vortex mixture or the vortexer. So this vortex mixture is a type of basic technology used for mixing of the which is used for the mixing of this sample in a glass tube of flask in laboratories. And the working principle is that it is based on a simple principle which causes the reaction and the homogenization by agitating the mixture. So this vortex mixture is commonly used for mixing of various sample fluids in the sample tube, and it also allows the homogenization of the cell and the cell organelles. Next we have here is the water bath. So this water bath is a conventional device that can be used for chemical reaction that are required for the controlled environment at a constant temperature. So this water bath is a device which transfer water temperature to a reference value, which is then amplified, and a control system generates a signal for the heating system, which heats the water to the desired temperature. So the main uses of this water bath are primarily it is used for heating the sample under a controlled temperature. Next we have here is the laminar airflow. So this laminar airflow is a closed device primarily for the processing of instrument that is sensitive to the microbial contamination. Here we can see the given picture of the laminar airflow, which is made up of a stainless steel, avoiding the joints and corners, that is to prevent the accumulation of the bacterial spores. And it creates a sterile environment with the flow of sterile air through a high efficiency particulate air HEPA filter and a shortwave ultraviolet germicidal lamp that sterilizes the workstation. So the main uses of this laminar air flow is commonly used to conduct the process that are sensitive to contamination. It is also used for experiment like related to plant tissue culture and the experiment of genetic transformation, and it is also used when the cultured media is prepared, or when we are performing any stain. It can be of gram stain or Zeldensen stain or a simple stain. So this is all about the instrumentation.